Welcome back to another class. Today we are going to look at human response to the environment, which is going to be dealing with the nerves, the brain, the spinal cord, the eye, and the ear. Let's go to our topic, the vertebrate nervous system. So we are talking about the human nervous system. The human nervous system is divided into three. The central nervous system, which includes the brain and the spinal cord. The brain is on the top, while the root of the brain is what you call the spinal cord. We are going to look at this in detail. The peripheral nervous system. Peripheral means sides. So this, if it is not central, then it means that it is on sides. So this central nervous system has side nerves, which we call the peripheral. That system which forms the peripheral nerves, we call it the peripheral nervous system. It covers all the body nerves. Then we have the sympathetic and parasympathetic, which we call the autonomic nervous system, and gland muscles. Smooth muscles are found in the walls. For example, in the walls of the intestine, and then cardiac found in the heart, and then glands are those small structures which produce hormones. So what is the function of the central nervous system? Number one, it allows the human to react to internal and external stimuli. If it's one, it's called stimulus. If there are many, we call them stimuli. So when you say it reacts to internal and external, those are, a stimulus is any change in the environment. For example, if the heat is low, then changes to high, that is a stimulus. So you have to respond from low heat to high heat. What do you do when there is high heat? So those actions you do as a result of detection in change they are being controlled by the nervous system. So internal changes could be like carbon dioxide, could be oxygen, could be level of glucose, and then external changes could be maybe temperature, it could be humidity, it could be sun, and others. Then it also controls and coordinates every movement and all organs, glands, blood vessels in a response to the information received. So the moment the body receives the information internally or externally, then the nervous system also helps in coordinating so that a meaningful reaction or effect is being done. Let's look at the brain. The brain has some specific parts which you need to know. We have cerebrum, we have medulla oblongata, we have pituitary gland, we have cerebellum, we have hypothalamus, we have corpus callosum. But these parts, they have specific functions they do. Cerebrum is the biggest part of the brain, whereby this cerebrum, it controls all voluntary actions. When you talk about voluntary actions, these are actions you do on your own will. For example, sitting, standing, raising the hand is an example of voluntary action. Then number two is receives and interprets sensation from the organs. When you talk about sensation, we are talking about the senses, like to see. Sight is a sense, smell is a sense, touch is a sense. All those senses are being controlled by the cerebrum. Then there is also a higher thought process. It's a center for higher thought processes. What are the higher thought processes? We have memory, judgment, and reasoning. So if you have any problem with any of those three processes, then it means that your cerebrum is having some problems. Then number two, we have corpus callosum. This one, it helps. The brain is divided into two, the left and the right hemisphere, and the left hemisphere controls the right part of the body, and then the right hemisphere controls the left part of the body. So these two parts are supposed to communicate, but how do they communicate? They communicate using this bundle of fibers, which we call corpus callosum. So it allows communication between the two hemispheres, the left and the right hemisphere. Then another big part of the brain is medulla oblongata. Medulla oblongata is very important. It's found here down towards the spinal cord. Sometimes students confuse medulla oblongata with the spinal cord, but the medulla oblongata is towards the pons. So medulla oblongata transmits impulse between the spinal cord and the brain. Since it is a junction between the brain and the spinal cord, so it allows the impulse, allows the information, electrical message to come from the spinal cord to the brain, and also controls involuntary. When you talk about involuntary, these are actions which you don't have control over. For example, here is an example of a heartbeat and breathing. You cannot say that my heart stop beating, even if some people say that when you leave me, my heart will stop beating. That is impossible, because the heart is not being controlled externally. It's being controlled by medulla oblongata. Then we have cerebellum. Cerebellum is very important for coordinating. Here it is coordinate, not control. It coordinates all voluntary movement. Don't confuse voluntary actions, voluntary movements, and involuntary actions. Voluntary actions, action you do on your own will. Involuntary actions, actions you do without your will. Voluntary movements, these are movements, and it is just coordinating them, like walking and running. Also controls muscle tone and maintains balance. So balance is being controlled by cerebellum. So it is not advisable to hit a student or a kid behind the head, because the kid might lose balance. And then also we have hypothalamus. We will see this one more when you go to endocrine. It controls, is a center for hunger. When you are hungry, is this. When you are thirsty, deep sleep, sleeping, body temperature, and emotions. All these are being controlled by hypothalamus. Then you have the pituitary gland. Pituitary gland is one of the very important gland which can secrete many hormones in the body. We will see this when you go to endocrine system. What are some of the questions? Here is the structure of the brain. Can you label it very well? Part A, we said part A is medulla oblongata. Part B is cerebrum. Part C is corpus callosum. Part D is cerebellum. Part E is spinal cord. Can you give the function of each? You have to be knowing those functions, so that you know that at least you have tried to master the brain. If you can't remember, then check up with your cerebrum. Give the name and the part labeled. We have seen the parts. How is the brain protected? It is protected by, number one, the skull, or what you call the cranium. Number two is also protected by the three main genes. Is not really needed to be known according to the examination guideline, but if you read other books, you can find them: the dioramata, arachnoids, and the piamara. Let's look at the nerve cells. Nerve cells. Nerve cells, there are three types of nerve cells. The first one is the motor neuron. The second one is in connector neuron, or what you call the intermediate neuron, and then the interneuron, and then another one is the sensory neuron. These three nerve cells, they have specific characteristics which you can use to differentiate among them. The first one is the cell body. The cell body of the motor neuron is found at the terminal, while the cell body of the interneuron is found along the axon, and the cell body of the sensory neuron protrudes out of the axon. You buy Thunder movies, you can contact them. And then also Royal College from grade R to grade 12. People around the Rodiport, you can contact them, grade R to grade 12, and also Randifield Education Center in Krugersdorp. Nerve cells are divided into three groups or categories. The first one, we have the motor neuron, we have the intermediate neuron, and then we have the sensory neuron. These three nerve cells, they are differentiated among them by the location of the cell body. In the motor neuron, the cell body is at the terminal, or at the beginning. In the interneuron, it is in the middle, or along the neuron, and then in the sensory neuron, it is protruding out of the axon. You can differentiate among them by the position of the cell body. If this is a motor neuron and this is a sensory neuron, the motor neuron, the cell body, we say that is at the terminal or at the beginning, and then sensory neuron is along the neuron. But what are some of the functions of these parts these neurons have? Number one, you have the dendrite. Dendrite is the part which brings impulse to the cell body. So this part brings the impulse from maybe the central nervous system to the cell body, and then you have the cell body itself. Cell body is this dense body here, which is containing the cytoplasm and the nucleus. It's the same thing here, which is the cell body. And then you have the nucleus. Nucleus contains the genetic material. Therefore it controls the activities of the cell. Then you have cytoplasm. Cytoplasm is where cell activity takes place. The axon is a part of a neuron which takes away information from the cell body. So this is an axon which takes away information from the cell body. Myelin sheath, when you say sheath, means something which is protective. So myelin sheath is very important in protecting the axon. So it insulates the axon so that there is no electrical shock. Remember, the impulse is transferred in form of electricity. So it is an electrical message. Since this is an electrical message and we have a lot of cells, then it means that each cell must be insulated. So the myelin sheath, it will take that function of insulating the cell. So this is a small description of what I've been trying to say. The nucleus controls the cell body, the cell's activity. The cell body consists of the cytoplasm and the nucleus. The cytoplasm contains the cell content and where the cell activity takes place. Then we have the myelin sheath. Myelin sheath, it covers and protects the axon. The axon conducts impulse away from the cell body. And then we have the dendrites. This one conducts the impulse to the cell body. So the axon is like the opposite of dendrite. The reflex arc. Reflex arc is, when you say arc, it means a path. So a path traveled by the impulse during the reflex action is what called a reflex arc. But what is this path? The path is being traveled by the impulse from the sensory neuron through the connector neuron to motor neuron and then to the effector. So when you say a reflex arc, it's just a path which the impulse is supposed to travel before an effect is being brought about. So here is the path. You have the receptor, for example the skin, the sensory neuron, the neuron or the nerve cell which picks the information from the skin, then to the connector, takes the information to the connector in the central nervous system, then the motor neuron. The motor neuron will pick the information from the central nervous system and then takes it to the effector, where the effect, like the muscles, where the effect is supposed to be brought about. Then the reflex action is a rapid automatic response to the stimulus. So the stimulus is supposed to be rapidly responded to. Any change in the environmental condition is what you call the stimulus. So any change, it could be harmful or helpful. Then you have the significance of the reflex action. What is the function of the reflex action? The reflex action is very important in protecting the body from further harm or from danger. Yes, the danger might be there already, or it could be approaching. Therefore, the reflex action will bring about in protecting the body from danger because of its rate at which it responds to the stimulus. Some of the examples include pricking the finger, knee-jerk reaction, removing the hand from the candle or hot object. All those are protective measures which protect the body from danger. You don't need to teach anyone to respond because then the answer is always negative. Even if it's a baby, when touches the hot object, the baby will not leave the finger there. You have not told the baby to take away the finger, but because of the hotness, the body is triggered so that the baby withdraw the hand. So reflex actions are always negative. Your answer is supposed to be getting away from the stimulus. The spinal cord. Let's look at the spinal cord. The spinal cord is being protected by the vertebra or the vertebrae layers. Is not basically needed in this scope or based on the examination guideline, but those who are interested, we have 3 of them, which is the pia mara, dura mara, and arachnoid. And also being protected by the cerebrospinal fluid. It has a fluid which prevents friction from taking place. Let's look at the structure of the reflex arc in detail. The reflex arc divided into sections, whereby you need to know all the parts of the reflex arc, the function, so that you can link them together to produce a meaningful message. So you have the receptor, it could be a skin. The receptor has sensory cells which sense the change in the environment. Those receptors convert the stimulus, that change the stimulus, into a nerve impulse, into an electrical message. Then the electrical message will travel along the sensory neuron. It enters the spinal cord via the dorsal root. Then the sensory neuron will connect to the connector neuron using a synapse. The synapse is a connection between 2 or more neurons. The connector neuron, or what you call the relay neuron, will connect to this motor neuron by another synaptic connection. The motor neuron will take the impulse away or out of the spinal cord via the ventral root. The motor neuron will take the information to the effector, in this case could be muscles or glands, and then the effector will react or will contract so that the hand or the part of the skin is withdrawn from the painful object. So you need to know how this mechanism brings about the information. So if you know the part and the function, then you'll be able to understand the mechanism. So it has what called the gray matter and then the white matter. It has the central canal. All those parts you need to also know them because they can ask you to level. So any action which moves out of the spinal cord and goes to the brain cannot be classified as a reflex action. Why? Because it requires some reasoning. In this case, you don't need to reason any. So if you look at this, it shows that you have the stimulus as the light, goes to the receptor as the eye. The eye will sense using its receptor cells, the retina, and then takes the information. This information will not take this route unless it is a protective measure to the eye, blinking of the eye. Yes, it will take this route. But if you to see the object, it requires some thinking, then automatically it is going to take this route and then goes to the brain, then comes back. Yes, they can ask a question and reply immediately. You see that that is a very short period of time, but it is very long because it requires processing of information in the brain. So the reflex arc is faster, the reflex actions are faster than those which are not reflex. So in the reflex actions or involuntary actions, you don't require any conscious. So the distance traveled by the impulse here and the distance traveled by the impulse here, this one is much more greater than the distance here. Therefore, the reflex action are very fast. That's why you call them, say, rapid. And you don't have control over them because you control it with the brain. Because it doesn't go through the brain, therefore it is involuntary. And those actions will help in protecting the body. But drinking water is not a protective measure. Therefore, it has to take this to say either you drink or you don't drink. Let's look at some of the questions. Can you answer these parts? We have A is the knee, we have C is the motor neuron, we have B is the sensory neuron, you have A is the connector neuron, you have D is the spinal cord. Can you know the function of them? Identify the neurons labeled A and B. A, we say that is the connector neuron, and then B is the sensory neuron. Write the letter and the name of the structure stimulated by the hammer. Letter is E, and then the structure is the knee or the skin. Remember, sometimes we ask you to give the letter and then the name. Sometimes just the name, sometimes just the letter. So you need to read the question before you attempt to answer it. Explain what would happen if C is severed. What will happen if C is damaged? It's no longer working. So since it brings information from the central nervous system to the effector, it means that the effector won't receive the information. But the sensory neuron will be working, it will be taking the information to the central nervous system. Therefore, this individual will be able to feel the pain, but won't be able to react. For example, you have a muscle pull, you feel the pain, but you won't react. It means that the sensory neuron is working properly, but the motor neuron is not working. What will happen if B is damaged and then C is working properly? Is this situation really happening in the life or their life? Yes. It means that you won't be able to feel the pain, but you will be able to react. For example, a person who has gone for removing of the tooth, the person cannot feel the pain when they are removing the tooth, but when they're asked to react or open the mouth, the person can do so. So it means that the sensory neuron is not working, but the motor neuron is working. That's why the reaction can take place. Then give examples of the reflex action other than the knee jerk. We say that blinking of the eye is also a protective measure. Withdrawal of the hand from the hot object is also another protective measure. Why are reflex action important? We say that they are always negative. So therefore they bring about the protection of the body from danger, since it is very fast in reacting. Then differentiate between the reflex action and the reflex arc. The reflex arc, we say that is the path traveled by the impulse in bringing about the reflex action, and then the reflex action is a rapid automatic response to the stimulus. So the reflex arc is path, while the reflex action is the action brought about by the impulse after traveling along the reflex arc. What is a synapse? A synapse is a junction between two or more neurons. So it is very important in bringing about the impulse to travel in one direction. So it allows the impulse to move in one direction. What are some of the diseases which affect the central nervous system? Number one is Alzheimer's disease. Alzheimer's disease is as a result of the frontal and the temporal lobes of the cerebral cortex being affected, and then here you lose the memory. So it means that the cerebrum, the part of the memory, is being destroyed. And then what happens? The patient becomes very forgetful as a result of the short-term memory centers being affected. This is being caused by genetics. It means that you inherit it from the parents. Number 2 is multiple sclerosis. Multiple sclerosis as a result of the myelin sheath surrounding the axon or in the white matter of the brain and the spinal cord being attacked by own immune system. So the immune system identifies the myelin sheath as the foreign object in the body, therefore tries to fight it. In doing so, it is destroying its myelin sheath. This could be leading to triangling, numbness in the limb, loss of coordination, weakness, and then chronic pain in the muscles, and can also result in depression. It is being caused by genetics. Yes, you can get it from your parents, environmental factors, the way how we live our life, and also infectious disease can also destroy the myelin sheath. Next time you're going to look at the human eye. How does it operate? Thank you very much. I'll see you in the next class. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Today we'll be looking at the human eye. All right, let's look at the human eye in detail. We are going to start with the part and its function, what we call the part and the function of the human eye. So in this case, sometimes it could be the adaptation of the eye for its function. It means that you are supposed to give the part and the function of the eye. Okay, let's look at number 1, which you call the cornea. So we are saying that it's very important in the refraction of light rays from the object. So when the light comes from the object and falls on the eye, the first layer to diffract this light, to bend this light, it will be the cornea. Then number 2 is the pupil. Pupil is this space between this iris and this iris. So this space is what called a pupil. So the function of the pupil is to allow light to enter into the eye because of the space which is there. So it means that light can go through that space so that it's being focused. And then you have the iris. What's the function of the iris? Iris is, because it is making this space, so it brings about control of the size of the pupil. It controls the size of the pupil. If you want to look at this one very clear, you can look at the cat. At night, you see that it has a very big pupil, and then during the day, the pupil is very small. So what causes that? It means that when the iris tries to pull away from each other, then the size of the pupil becomes big. Then when it tries to come next to each other, yes, then it means that the size of pupil reduces. So when the pupil size increases, it means that more amount of light will enter the eye. Then you have the lens. This lens, this solid substance is called a lens. The function of the lens is to changes the shape for near and far objects. So it is very important in focusing the near and far object. So it means that if you are not able to see near and far objects, you can see near or you can't see far, then it could be a problem coming from the lens. Then you have what called a scleroid. This one sometimes called a sclera. It is very important in protecting the eye against damage. So it means that if you look at it, the eye in the inner part of the eye is being protected by the sclerotic membrane. Then you have another part which called the choroid. This is the dark part of the eye. This part is dark, yes. It's very important in reducing refraction. So why is it important? When the light rays, yes, come, yes, sometimes some of them they are focused to the retina, yes. When the light rays come, they are being focused on the retina. So some of it, like it tries to go through. So it should be reflected back. It's supposed to be reflected back. So the function of this choroid is it tries to absorb that light so that it's not being reflected back to or bring about different images in the eye. Then you have the retina. This is where the image is formed. Basically, retina has the sensory cells which can detect during the dim light and the bright light, which you call the cones and rods. So the cones are very important even in seeing the color, while the rods are they cannot see the what they cannot detect the color. So basically, cones are frequently used during bright light or during day, and then the rods are being used more during night. It means that nocturnal animals like dogs, like lions, the one which hunt at night, their rods are more advanced or they are more developed compared to the cones. Why? Because they can't see the color, but they can see at night better than seeing during day. So rods are very important in sensing small amount of light. So small amount of light falling on it, it can be sensed. So you have another part which called the yellow spot. Yellow spot is where the clearest image is formed. So this part which bends outwards, it is where the clearest image is formed. Sometimes when you are looking at, you tilt your head so that you can focus an individual nicely, or you can see the individual clearly. So it means that you're trying to make the image to fall on the yellow spot. Sometimes we call it fovea. Sometimes we call it fovea. So this is where the clearest image is formed. Then you have what you call the optic nerve. This one carries impulse from the retina to the brain. When we talk about the impulse, we are talking about an electrical message. An electrical message. So when light falls on the retina must change this light into an electrical message. That electrical message is being carried to the brain in the form of impulse. So therefore, it means that an impulse is an electrical message. So optic nerve, they are carrying this impulse to the brain. Whenever you talk about optics, it means that you are talking about light. Then you have what you call the blind spot. Blind spot has no vision. Why? Because we don't have any cells there which can detect light. So whenever light falls on the blind spot, you won't be able to see. Then you have the suspensory ligaments. This one holds the lens in the position. So if this is the lens, you can see that these small threads are holding this lens so that the lens can be in position. Then you have the vitreous humor. Vitreous humor is very important in giving the eye its shape. So this fluid which is found in this eyeball, so it prevents compression. So because it is a liquid, so it prevents compression, and then the eyeball can obtain its shape. Then lastly, we have what called the aqueous humor. Aqueous humor is found in front. It's very important in supporting the cornea and also the front chamber of the eye, because it's also like fluid-like. So it brings about part of the eye so that it can stay, everything can stay in what? It can stay in shape or in position. And also it balances. If this side there is a pressure, then this pressure on this side should be balanced by this fluid this side. So basically, that's some of the functions of the eye. If they ask you the adaptation of the eye, it means that you have to look at one part, then give the function. Give the one part, and then give the function. So please don't go to the exam without knowing the functions of each part, because you don't know which part they might bring if it comes. Let's look at binocular vision. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. When you talk about pupillary mechanism, it's the process by which the diameter of the pupil is being changed. You find out that students tend to misunderstand the pupillary mechanism and the accommodation. Basically, in accommodation, you have to talk about the lens, you have to talk about the suspensor ligament, you have to talk about the ciliary muscles. While in pupillary mechanism, in this case, you talk about the muscles in the iris. And you also talk about the pupil. How much light is entering the eye? That is what you call pupillary mechanism. So it's the process by which the diameter of the pupil is altered to control the amount of light entering the eye. How much light is entering the eye? So in this case, the light can be changed depending on bright or which is in dim. So during bright light, then it means that more light is entering. And then during dim light, it means that less light is entering. So now the eye is supposed to adjust so that at least it's the same amount of light which is entering the what? Which is entering the eye. How does that eye do this? During bright light, the radial muscles of the iris relax. And then the circular muscles of the iris contract. When they contract, the pupil constricts. It means that it gets smaller and then less light enters the eye. How do we explain this? Why are these ones in different color? I can say that R goes with R, C goes with C. R with R, C with C. What does it mean? I mean this. R, which is radial, goes with relax. And then C, which is circular, goes with contract. Radial muscles relax, and then circular muscles contract. Simple. If they contract, then less light enters the eye. That's what we need from there. So if you look at circular muscles, they call them circular muscles because they're in circulars. They're like in circles. So that's why it's called the circular muscles. And then radial muscles, it's like a radius. So that's why it's called radial muscles. When the radial muscles relax, when these muscles relax, then the circular muscles will contract. When this one will contract. When they contract, what happens? It means that the diameter of the pupil will decrease. And then when it decreases, it means that less light will enter the eye. So it means that it's pushing this into the middle so that now this hole is what? Is small. And then less light will be entering the eye. So that is R with R and then C with C. You just need to know this. Radial muscles relax, circular muscles contract. Less light, the diameter of the pupil reduces and then less light enters the eye. Simple. That is bright light. What about dim light? In dim light or dim conditions, what happens? The radial muscles will contract and then the circular muscles will relax. It's just the opposite of the other one. So the pupil dilates. When you talk about dilation, it means that it becomes bigger. It increases in size. It becomes wider. And then more light will enter the eye. So what does it mean? I mean that now, if you compare this one with the other one, compare this one with the one in bright light, you find out that the pupil here is big. Even this distance, you see that it's big enough. So what does it mean? So I can abbreviate it that radial with the contract and then circular with relax, which means that R with the C and then C with R. I tell my students, I'm bright like a star. I don't mix things up. When you mix things up, you mix things up in dim, in darkness. It's when you mix things up. So in bright light, I don't mix the things up. So the R will go with the R and the C will go with the C. While in dim light, because I mix them up, R will go with the C and then C will go with the R. That's the meaning. So it shows that during dim light, we require more light to enter. That's why now the pupil is supposed to become bigger, or it must dilate so that more light can enter to maintain the amount of light, how much light is entering the eye. So it must be kept constant. That's why when there is too much light outside, then it means that the eye must constrict so that less light is entering or the same amount of light is entering. So basically, if you look at this, you can compare now them. During bright light, it looks like this, the pupil is small, while dim light, the pupil is big. You see it diagrammatically. If you look at it diagrammatically or structurally, you'll find out that these ones, the spaces here and the spaces here are not the same. You see that here, the space here, the amount of light entering here is little, or the space where light is passing is small compared to this. So I'll say that this one has constricted while this one has dilated. So basically, that's what you need to know during dim and bright light. During dim light, more light enters the eye, while during bright light, less light enters the eye. What are some of the eye defects? We have short sight. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Let's look at binocular vision. So is a vision using two eyes with overlapping fields of view, allowing good perception of depth. It means that you can judge exactly in which distance and how big is the size of an organism using these eyes. So it means that you have multiple images which are being formed, but the brain is supposed to interpret it as one organism or one individual being viewed. So if you look at this, you see that a lot of light is entering with different light rays, but when they enter, because I have one eye here and I have another eye here, it means that I have different images which are formed. One image from this side, for example, and one image from this side, for example, and then when they are being sent to the brain, it is being interpreted as one object. So binocular vision is very important. It's very important in developing good perception, in developing good depth perception, which in turn affects the coordination and the hand-to-eye skill. It means that if you're using two eyes, if you close one eye and then you try to do a target, yes, of, for example, a small hole, then you won't be able to do it. So it means that if you use two eyes, then you can judge the actual distance. For example, catching a basketball, making sense of classroom instruction on the blackboard or screen, and then performing simple tasks such as walking and running. So you require, it means that a person who is having two eyes is more advantageous. So that is binocular vision. So this makes us to go to what you call accommodation. Accommodation, when we talk about accommodation, we don't mean the accommodation where you stay, no. Here is like ability of the eye to see near and far object. So it means that it's supposed to change the focal length of the lens so that it can see the near and far object. So it's the process of changing the shape of the eye lens to focus the near and the far object. So it means that the eye is supposed to change its shape. Basically, the lens is supposed to change its shape so that it changes the focal length. If the focal length is changed, then it means that the distance where the image is being formed behind it also changes. So it means that now the eye will be able to see the near and the far object. So let's look at the near vision. When you talk about near vision, you're talking about an object which is less than 6 meters from that object. More than 6 meters from that object, then it means that that is a far object. So what happens during near vision? One, there are some muscles which you need to understand in this case so that you can understand how it operates. And then number two, when you are trying to describe near vision, when you understand the near vision, the far vision, it will be just the opposite of the near vision. So there are some muscles which call the ciliary muscles. And then there are other muscles which you call the suspensory ligaments. Then you have the lens. Yes, you have the lens. And then you can talk about the light. So let's try to describe this. So if you say that ciliary muscles, if these are the ciliary muscles, yes, if this contract, then this one must relax, or they must loosen. So they work antagonistically in the opposite way. So ciliary muscles will contract. When the ciliary muscles contract, yes, if the ciliary muscles contract, then this one must relax because they work in the opposite way. Suspensor ligament must slacken. It means that they must become loose. And then because they are loosened, then it means that this one, the tension on the lens has decreased. The lens become more convex. It means that light rays are refracted more because it is coming from a big object or a nearby object, so it looks to be big. So light rays are being reflected more, and then the clear image is being focused on the retina. Let me give you an example. If you are viewing an object which is near, so it means that that object, you see it as a very big object. So it means that it will require a big lens, yes, or a thick lens, or a more convex lens, so that that lens can bend this light rays, yes, which has come with a big field of view onto the retina. So if the lens was small, then it means that that power of the lens is not going to be enough to bend this light onto the retina. Therefore, the lens must be big. So when you're explaining this, how do I know that the suspensor ligament must relax or slacken, and then ciliary muscles must contract? If the lens is big, so if you are viewing an object which is near, then it means that the lens must become big. Near object looks big, therefore the lens must be big enough to bend this light from this object. Therefore, if the lens is big, it means that it's more convex. Then it means that what is attached to the lens must relax, or it must slacken, which is the suspensor ligament. And if the suspensor ligament slackens, then it means that the ciliary muscles must contract because they work antagonistically. From there, now you can start. The ciliary muscles contract, the suspensor ligament becomes slackened. When they slacken, the tension of the lens decreases. The lens becomes more convex, and then the refracting power increases. Light rays are bent to the retina more. Light rays are being bent more, and then the image, a clear image, is focused on the retina. That's how you can try to explain that. Then if you look at the distant object, it's just the opposite of the near vision, more than 6 meters from the object. So in this case, we will see that it's just the opposite of the other one. The ciliary muscles relax, the other one contracted. Then suspensor ligaments become tightened. It means that the other one is slackened. It becomes taut. It becomes tightened, like becomes stiff. So the other one was slackened, like relaxed. The tension on the lens increases. Then it means that the other one, the tension on the lens decreased. And then the lens became less convex. It means that it became small. And then the other one, the lens became big. If you do the tension on the lens becomes less convex. And if you release the tension on the lens, then the lens becomes more convex. That's how it works. Then you're saying that light rays are being reflected too. Are being reflected less. Why? Because the lens is less convex. And then clear image is being focused onto the retina. So this is how it works, that if this one becomes, it relaxes, this one must contract, and then this one becomes, if it contracts, it means that this one is pulling this side, and then also this one is pulling this side, and then the lens becomes small, which means that the lens becomes less convex. And then because it is less convex, thickness is too small, it means that its refracting power is small, is less. Therefore, it means that the light rays are not going to be bent as much as like that one of near object. And that's why when you are seeing the object which is far, you see it as small. Why? The light rays come as a very small beam, and then it is being bent to the retina, and then a clear image is being focused there. So that's how you can differentiate between these two. What about pupillary mechanism? Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. We have short-sightedness. What does it mean? Short sight. It means that you can see objects which are near in short distance. Short sight. That's why it's called short-sightedness. Then it means that you can't see objects which are far. So it's an ability, is the ability of the eye to see nearby object, but cannot see objects in a distance or objects which are far, more than 6 meters from the individual. This defect may be caused by, number one, eyeball is too long. It means that the eyeball is too long, whereby the image is formed in front of the retina. The cornea is being too curved for the length of the eyeball. Then it means that if it is too curved, then it means that the light is being bent too much compared to how it's supposed to be bent. Image is then forms in front of the retina. And then inability of the lens to change, inability of the lens to become less convex. So if it becomes less convex, then it means that the object, the light will be bent more, and then it forms in front of the retina. So the normal vision, the image is supposed to form here by the retina. But because now, number one, the eyeball is too long, this eyeball is too long, so the image does not form on the retina. It can't move up to the distance of the eye to finish this distance. So it forms in between. The focal length of this lens is not by the retina, is in between here, and then the image falls in between. Oh, the cornea is too curved. It means that this cornea in front here, it bends this light more. By the time it falls on the lens, it's already bent too much, and then it ends up forming the image in between. Or inability of the lens to become less convex. It means that the lens remains big, and then refracts this light more with a high power, and then it forms the image in between. It does not reach the retina. You can treat this defect by wearing glasses which are concave. It means that at least you put a concave glass here which diverges this light so that it appears to be like an object coming from near object. And then it means that this light rays will be bent to, it means that now this image will be shifted to the retina. Then you'll be able to see the object. This is what I'm trying to say, that you put what you call a concave lens. This concave lens tries to bend light and then shifts this image which is formed here to the retina. Now the image will be formed onto the retina. Then number two, we have what you call the long-sightedness. It means that you can see objects which are far, long distance, but you can't see objects which are near. It's the ability of the eye to see distant object, but you can't see nearby objects. The defect may be caused by, number one, the eyeball is too short. It means that the image is going to form behind the retina is too short. Yes, the distance supposed to travel. The focal length of the lens is going to be behind the what? The retina. And then the image is formed behind the retina. Number two, the cornea is being less curved. It means that the length of the eyeball is what? Is short. And then it means that the image is being formed behind the retina. Then inability of the lens to become more convex so that it can bend this light to the retina. So it means that its power is less. It can't refract this light onto the retina. So it just have to pass out. It's like if you have the stopping, you are supposed to stop at a certain point and you are running, because the speed is too much, yes, you end up bypassing that point and then you stop after the point. So that's how it is. It means that the power of this light is too much. Therefore, the lens fails to stop or to bend this light to the retina, and then the image ends up forming behind the retina. Then if you look at the picture here, it shows what really is happening. This is the normal vision. The image is formed in the retina, but on the long-sightedness abnormal eye, or eye with the defect, the image is formed behind the retina. So our job now is to bring this back to here. So what do we do? We need to reduce this beam, so the wideness of this beam, so that the image is brought back to the retina. So how do we treat this? We treat it by wearing what you call the convex lenses. What do? Convex lenses, they bend the light. If it is convex, it means that they bend the light. It becomes, it converges the light. By the time it reaches the lens, then the light has been converged. Then the third one is what's called astigmatism. Astigmatism is when the cornea of the lens, is not equally round in all directions. Light is not focused evenly on the retina. Then it means that you'll be seeing multiple images. This leads to, number one, blurry vision. Number two, headache. It means that you can't see. It's like having a phone and video calling someone, and someone is shaking the phone. So it becomes, you start to feel that headache. That's what happens to a person who is suffering from astigmatism. Then you have the squinting of the eyes. So what happens? It means that the eyes are not proper in the socket. Person who is suffering from astigmatism has multiple images are being formed. So now the brain becomes confused which image is to be interpreted. However, it's going to interpret everything, and then it's going to bring back the effect. You'll be seeing multiple images. That's why you have what you call the blurry vision. Treatment. How do you treat this? Number one, you can treat it by putting on glasses with prescription lenses. It means that it depends on the severity of your astigmatism. Then the prescription of lenses will be given to you. Then contact lenses. It means that you can put it on your eye so that it can change, or it can bring about the normal focusing of the image. Then lastly, laser therapy. Then it means that you can cut off that cornea, or you can scoop it out so that it becomes evenly distributed. The last one is what called cataracts. Cataracts is like milk substance in your eye. It occurs when a clear transparent lens becomes cloudy and opaque. It means that it does not allow light to go through, resulting in a blurry vision. This is this is the normal eye, and then this is the person with the cataracts, how it looks like. It clouds, it becomes like milk, and then it solidifies. The lens solidifies, and then you won't be able to see. So it means that the lens cannot even change its shape. Why? Because it has solidified. How do you treat it? Treat it by doing a surgery, or surgery to replace the lens with the synthetic lens. It means that we remove the lens which was there, and then we put the one which is synthetic, which means that the one which is manufactured, man-made lens. Here are some of the questions you can try and then do them. You can pause the video and then you do them, and then see if you can get the correct answer. I'm not going to discuss them, but you can pause the video, you do them. So these are some of the answers for the basically yes. So if you have any question related to life science, you can write it in the comments under the video, or you can WhatsApp us, then we'll be able to help you out. The human ear. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Let's look at the human ear. Under the human ear, we have to look at the different parts of the ear and the functions, how the ear functions,
It means that that is the hearing process. The defects of the ear, meaning that people having problems with hearing. The ear is divided into three parts. The outer ear, this part, we call it the outer ear, consists of the pinna. The pinna is this flappy part which you see outside. So it's called the pinna. So the pinna is connected to what called the auditory canal, and then it also has what called the tympanic membrane. When you're marking, we call it tympanic membrane. We don't call it eardrum. We prefer calling it tympanic membrane. So it has the pinna which collects sounds and then sends the sounds to the auditory canal. Then the auditory canal has to bring the sound to the tympanic. So now it has what you call the middle ear. The middle ear is made up of these parts, whereby it has the three bones, which we call oscars, the malleus, the incus, and the stapes. And then it has what you call the eustachian tube. Eustachian tube is that tube which connects to the mouth. So like when you yawn, you feel like some air is going through the ear because of the eustachian tube is connected. Yes, the mouth is connected with the eustachian tube. So basically, this is the oval window. Yes, and then it's like an oval shape. That's why it's called the oval window. Then this one is round. It's a round shape. That's why it's called the round window. Then you have what you call the inner ear. The inner ear is divided into different parts also, whereby you have the cochlea. Then you have the auditory nerves. The cochlea is where you find the cells detecting the sound or the vibration. The auditory canal is those ones. Auditory canal, they send impulse to the brain. And then you have the other part of the ear, which is responsible for balance. So we call it the semicircular canals. The semicircular canals. So basically, these are some of the parts which we need to look at in the ear. Then we need to know the function of each so that we can bring about the functioning of the ear. So we need to look at the functions of each part so that we can bring about the normal functioning of the ear. So the first part is what you call the pinna, which is the outside part of the ear. It collects and directs sound wave to the tympanic membrane. It means that when it collects, it has these curves so that it can collect sound wave from any direction and then sends it to the tympanic membrane via the auditory canal. So now the auditory canal, it transmits sound waves. So when the pinna collects the sound, it gives it to the auditory canal. When the pinna collects the sound, it gives it to the auditory canal. The auditory canal sends the sound wave to the tympanic membrane. Then the tympanic membrane transmits the sound vibration to the middle ear. So when the sound wave hits the tympanic membrane, the one which we call the eardrum, but we prefer calling it the tympanic membrane. So this, it vibrates. It's like beating the drum. When you beat the drum, it vibrates and then the sound is being produced, causing these three bones to change the position. During the changing of the position, then it means that the sound is being transmitted in the form of vibration. So we have the oscars, these three bones, which call the malleus, incus, and stapes, which we call MIS, M-I-S, MIS, malleus, incus, and stapes. Hammer, anvil, and stirrup, we basically don't advise to use it in the exam, although it is correct, but we don't advise it to use in exam. We prefer using malleus, incus, and stapes. These bones, they amplify the vibration. They amplify the vibration and also takes the vibration to oval window. So oscars amplifies the vibration and also transmits the vibration to oval window. Basically, that is its function. And then we have semicircular canals. These ones, they have sensory receptors for balance. That is the cristae and maculae. Then you have these wires, which you call the nerves. Yes, the nerves, they are very important, transmitting the impulse to the brain. So some come from the semicircular canal, responsible for balance, some from the cochlea, which is for hearing. So both of them are being transmitted to the brain. However, they go to different parts of the brain because balance is controlled by a different part of brain from hearing. So hearing is a sensation which is being controlled by cerebrum, while balance is being controlled by cerebellum. Then you have another part which called the cochlea, the coiled part. So when you're talking about hearing, you have to uncoil it. Then it contains the organ of corti, which converts the sound waves into the impulse. So the organ of corti has the sensory cells which detects the change in pressure. They're being stimulated depending on how much pressure is being applied, and then it converts that pressure or waves into an impulse. The impulse is being sent to the brain for interpretation. Then you have what you call the eustachian tube. Eustachian tube is very important in equalizing the pressure on the either side of the tympanic membrane. So what is the tympanic membrane? Is this. So if there is a pressure on this side coming in, and then this pressure is supposed, it means that there will be high pressure this side and then there will be low pressure this side. Then it means that this eustachian tube must bring the pressure, so that now the pressure inside and the pressure outside is equal, so that there is no rupturing of the tympanic membrane. And if there is low pressure on this side, then it means that also this side, it must be lowered, so that now the pressure is equalized here. But if the pressure one side is greater than another side, then it might cause the tympanic membrane to rupture. Therefore, eustachian tube is very important in protecting this tympanic membrane by equalizing the pressure on the either side of it. Then you have what called the round window. Round window is very important. Yes, we have also pressure inside the inner ear. So the round window is very important in absorbing that pressure from the inner ear. Remember, when the vibration comes, it enters into the cochlea. Some pressure is supposed to be sent back because not all of it is being used, and then that pressure is being absorbed by the round window. So the round window is very important in raising the pressure from the inner ear. So it absorbs the pressure from the inner ear. It absorbs pressure from this part of the ear. And then lastly, the oval window transmits the sound vibration to the inner ear. So the vibration which is caused, so oval window is very important in transmitting the vibration from the middle ear to the inner ear. So basically, this is what you need to know about the ear, the functions of the ear. Please know the part and the function, So how is the outer ear adapted its function of collecting and bringing the information to the inner ear? How is it? Therefore, you just have to look at one part and then talk about the function. One part and talk about the function. Let's look at the hearing process. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Let's look at the hearing process. Before you talk about the hearing process, you need to look at how the ear receives and takes the information to the brain. So you see, this is what you're going to describe during the hearing process. Sound waves are being received by the pinna. Then the pinna sends the information to the tympanic membrane via the auditory canal. Then the tympanic membrane causes the three bones, the oscos, the malleus, incus, and stapes, to vibrate. The vibration is being amplified, and the last bone, which is the stapes, causes the oval window to vibrate. The vibration is being transmitted into the fluid or the inner ear, the fluid. So the oval window causes the fluid in the cochlea, that is the perilymph, to vibrate. as it vibrates or as it's making a wave, some of the wave or the vibration enters the endolymph. This is the endolymph. It's called a peri because it's outside. It's called the endo because it is inside. So it's peri because it is on the sides, the peripheral sides of the cochlea. So that's why it's called the perilymph. So the outside fluid is called the perilymph. The inside fluid is called the endolymph. So the perilymph vibrates. Some vibration enters the endolymph. Inside the endolymph, you have this line which has the sensory cells. So those sensory cells are being stimulated. Some pressure goes through this membrane and then it's being absorbed by the round window. That's why we say that the round window absorbs the excess pressure from the inner ear. So basically that's how you need to explain. Let's look at it. So number one, we see that the pinna traps the sound waves and directs them to the auditory canal. That's what I've said, that it traps and directs it. So the major key, the major points here is traps and directs it. The sound waves to the tympanic, traps and directs the sound wave into the auditory canal. So the tympanic membrane vibrates. This causes the tympanic membrane to vibrate and transmits the vibration to oscars. Those are the three bones in the ear. The oscars tries to the amplification and then carry the vibration to the middle ear via the oval window. Then the oval window vibrates and it causes the pressure wave in the inner ear. That is basically the perilymph, as we saw it. Then the cochlea, that is inside the inner ear, vibrates and the sensory cells in the organ of Corti are stimulated. It means that now they change this vibration into a nerve impulse. And then the impulse is being sent to the brain via the auditory nerves. So the auditory nerve transmits the nerve impulse to cerebrum for interpretation. And then now we can now react according to the need or according to the interpretation of the brain. So that's it. And this is the demonstration. This is the direction. So when you're explaining hearing, you need at least to master the part and the function of the ear. Then you will be able to understand the concept. Then let's look at some of the questions which can be asked. Here is a diagram saying that give the letter a name. When they say letter, the letter which is being given on the diagram and the name of the part that transmits impulse to the brain. So it means transmits impulse to the brain. So you see, you have to be knowing the part and the function, which is F, and then you tell us which name is that. This is auditory nerves. Then allow pressure to equalize between the outer ear and the middle ear. Allow pressure to equalize between the outer ear and the middle ear. So it means that it equalizes pressure on the either side of the tympanic membrane. That's the part they are looking for. So the answer is, so it's G and then eustachian tube. Give only letter of two structures in the diagram of the ear that, when damaged, would result in the following. Number one, conductive hearing loss. And then sensorineural hearing loss, B and C. B is being caused by neurons. It's basically about the senses. So it's about E and F. While conducting is like a material which conducts the vibration, So it's gonna be B and C. Middle ear infections are a common cause of hearing loss. State one way in which middle ear infection are treated. You can treat it using antibiotics or you can treat it using grommets. We're gonna see them. Name the part of the ear where earwax is produced. That is cerumen. Sometimes we call it cerumen. That is the auditory canal. Explain why hearing loss is due to wax is usually temporal. It means that you can remove the wax and then you come back to the normal hearing process, or you can hear them nicely again. These are the answers of what we have tried to explain. Let's look at balance, the process of balance. Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. Balancing in human body. Balance, you need to know some of the concepts about balance. Number one, you know about the stimulus. There is speed and there is direction. Speed and direction, that is one stimulus. And then there is position of the head, it's another stimulus. So you need to know the receptor, which is the cristae and the maculae in this regard. Then you need to know the location of these receptors. Where are they located? In the ampulla of the semicircular canal. Then this one is located in the circulus and utriculus. And then you also need to know that when they change the stimulus into an impulse, then it's being sent to the brain. Which part of the brain? Cerebellum for interpretation. Remember that hearing is being sent to cerebrum, but this one goes to cerebellum for interpretation. So how do we explain this? So we are saying that change, now you're writing what is needed, like for example in a paper. Change in the speed and direction of the head, comma, stimulates the cristae found in the ampulla of the semicircular canal, comma, change in the position of the head stimulates the maculae found in the circulus and utriculus. The stimuli is being converted into impulse, and the impulse is sent to the brain along the auditory nerve for interpretation. The stimulus is sent to cerebellum for interpretation to restore balance along the auditory nerves. So I repeat, change in the speed and direction of the head stimulates the cristae found in the ampulla of the semicircular canal, comma. Change in the position of the head stimulates the maculae found in the circulus and the utriculus of the semicircular canal. The stimuli is being converted into an impulse, which is sent along the auditory nerve to cerebellum for interpretation to restore balance. Simple. What else do you need from that? Nothing, and that's what we need. So when you're writing, at least know how the flow. Sometimes they just bring a question whereby you just have to explain change in the speed and direction without the position of the head. Then it means that now you just take this route until you reach here. If they just bring position of the head without speed and direction, then you just take this route until you reach here. If they bring it, the question is open, then you have to take this route and you take this route, then you combine them until you reach here. They have to ask questions, you know. There is no way they won't ask questions outside the scope. So summary of balance, that is what I've been trying to explain. The cristae in the semicircular canals are stimulated by change in the speed, by change in the direction and the speed of movement. They are saying that macula in the semicircular canal and utriculus are stimulated by change in the position of the head when stimulated. Then what happens? This cristae and the maculae, yes, they convert the stimuli received into a nerve impulse or into nerve impulses. And then when they are transported along, which are transported along the auditory nerve to cerebellum for interpretation. so that cerebellum then sends impulse to muscles to restore balance. That's it. Then you get your marks. Just get your marks. Don't need to waste time writing a lot of things. This is what we need. Make sure that what is highlighted, you don't miss them when you're writing your essay or when you're writing your question. Hearing defects, problems with hearing. So we have number one, the middle ear infection. This is whereby excess fluid in the middle ear caused by pathogen infection. When you say pathogen, a pathogen is a disease-causing organism. For example, bacteria, viruses. Yes, it's an organism which causes diseases is what called a pathogen. So we are saying that excess fluid in middle ear caused by pathogen infection, specifically bacteria. Then how do we treat it? Because it's bacteria, then you have to apply antibiotics. So number one, because there is a fluid which has been formed, so we need to drain that fluid out. Therefore, we have to insert what we call a glommet. So you insert glommets. Number two, antibiotics. Antibiotics are for bacteria. So when you apply the antibiotics, then it means that the bacteria will be killed. This is how it looks like. This is a grommet. And then we insert it so that now the fluid can be drained. So we bring this one, we put it there. So let's magnify it and then you see what is really happening. So when this one, you bring it here, when you magnify, this is the tympanic membrane. So this is the grommet. Now the fluid starts to come out of the tympanic membrane. And then you are able to drain it out. Then your ear will become fine again. Number two is deafness. Deafness, it might be partial deaf or it could be total deaf. Whereby, if it's partial deaf, it means that you have difficulties with hearing. Total deaf, it means that you don't hear at all. It is caused by, number one, injury part of the ear, nerve, or parts of the brain responsible for hearing. So it means that now the brain can't interpret what is being received. Then hardening of the ear tissue, such as the ossicles. It means that the ossicles cannot move, cannot change their position. Then it automatically, the vibration cannot be taken to of a window, so that is taken to the inner ear to be detected by the organ of Corti. Then treatment, how do you treat it? By using hearing aids. Hearing aids, like help, you try to improve your hearing. Yes. Number two, cochlear implant. So it depends on which part of the ear has been damaged, then it will apply whether use a hearing aid or use the cochlear implant. This is what we call the hearing aid. Yes, it's like a microphone. So it's just aiding, it's helping you just to improve the hearing. And then the cochlear implant, they have to do a surgery on you. They have to cut, they have to do a surgery on you, they insert it. It means that now your ear is no longer used at all. So this is what you call the cochlear implant. This is just a tattoo, just trying to resemble the key. Thank you very much, guys, for watching our classes. We will be looking at other topic. Subscribe for new videos every single day. We publish videos to help you pass. We are looking for a way forward to make you pass and succeed in your life. Remember, Mandela said, welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. We are saying that genetics, when we talk about genetics, we are talking about what we call genes. Small sections of DNA and these sections, they have a certain character they are controlling, is what you call genes. So the study of inheritance, the study of these genes is called genetics. Basically, in this topic, these are the things you need to cover. I'm going to cover the following subtopics in this topic. First one is concepts of inheritance, the terminologies. You need to know the terminologies. Monohybrid crosses, crosses which involve one character, mono, one character. Sex determination, what is the percentage chance for you producing a girl or a boy? How do you determine the sex of a kid? Sex-linked inheritance, the characters which are linked to the sex. Blood grouping, which type of blood group, and then inheritance of blood group. Dihybrid crosses, when we talk about dihybrid crosses, we mean inheritance of two characters. Di means two. Genetic lineages or pedigree. These ones are tracing characters in your family. In most cases, in this case, you're going to be looking at the disorders which can be inherited by the family. So you have mutation. When you talk about mutation, it means change in a part of DNA. It could be the whole chromosome can change, or it can be just a small section of DNA changing. So we're going to look at the mutations. Genetic engineering. Genetic engineering, it means that you're trying to manipulate. So you try to manipulate with the genetics or with the genes of an organism. Then you have a paternity test. Here you're trying to do the testing of the parents, father, mother. If there is any subtopic you don't understand, then it means that you are still lacking, you are susceptible to lose marks because you don't know which part of genetics they are going to bring. You can bring any part from genetics. So you don't know exactly which part they're going to bring. And if you don't study that part, then it means that these marks which are allocated on genetics, you have lost some marks even before you go to the paper. However, by chance, you can find out that the part you don't know, it has not come, and then you can answer those which you know. But I don't encourage that. So let's look at the introduction. Father of genetics. When we talk about father of genetics, we are talking about Gold Mendel. He did a lot of work on inheritance, and he discovered a law which we call law of inheritance. He concluded a gene come in pairs, like when they are, for example, height is being controlled by two parts of genes. When we talk about genes come in pair, a character like, for example, height, size, skin color, they're being controlled by two kinds of genes. So they come in pairs and are inherited as distinct units. It means that one character is not interfering another character during inheritance. Each character is inherited differently, from one from the father and one from the mother. It means that each parent is supposed to give one part of the character. Remember, there are supposed to be two, so it means that the first part comes from the father, the second part comes from the mother. That's what he is saying. So let's look at some of the concepts of inheritance, meaning that the terminologies. So chromatin. When you talk about chromatin, it's a substance within a chromosome containing DNA and a protein. Basically, when you talk about a chromatin, chromatin is DNA. But in this case, this DNA is like threads. Yes, it is in coiled form. When it coils, then it forms what you call a chromatin. Then number two, you have what you call chromosome. Now, when the chromatin condenses, when it shortens, when it becomes thick, it forms what you call chromosome. So chromatin and chromosomes, they are the same thing, but in different versions. So you can say water in ice form and in liquid form. So it's the same thing, but it's in different phases. So we are saying that thread-like structures of nucleic acids and are found in the nucleus carrying genetic information. So chromosomes are the thread-like, they are like threads crossing each other. And these chromosomes, they carry what called the genetic material. You look like your father because of the genes your father gave you. Then, if you look at this structure, look at this, you might think that this is noodles. Yeah, you think that these are noodles or spaghetti. Yes, but if you condense it, yes, so if you shorten it, you squeeze it, you condense, it forms what you call chromosomes. So when they cross, they look like this. So when you talk about chromosome and the chromatin network, they are the same, but in different phases. You see, you can see that what is here is almost what is here. Small ingredients just to make them to be in this shape are being added, just to make them to be chromosomes. But what they carry inside, the most important thing, what do they carry, is the same information as chromatin network. But they form chromosomes during cell division. What about genes? Chromatin network, chromosomes, genes. Genes are just small sections on the chromosome. So if you just break down the chromosome or the DNA, you form different portions. Those different portions, they form what called genes. And then you have, we are saying that this is a small portion of DNA coding or controlling a particular character, a particular protein. So it must be these characteristics you have. You are dark-skinned, you are light-skinned. What causes that? It is genes. And then when you talk about allele, alleles are genes. Alleles form chromosomes. Alleles form chromatin network. But chromatin network, if you look at chromatin network, you look at chromosomes, you look at genes, you look at alleles, they are the same things. All of them is the same thing, but at different scale. I'm gonna give you an example. When you talk about that, where do you stay? You say that I stay in South Africa. Where exactly? Then you tell me that I stay in Northwest. Where exactly in Northwest? You tell me that I stay in Rustenburg. Where exactly in Rustenburg? So you keep on reducing the scale. Chromosome, genes, alleles, they are the same but at different scales. So when you talk about allele, it's an alternative form of a gene. You can be tall or short. So an alternative form is called an allele. So we are saying that alternative form of a gene found at the same locus. Locus means the location of a gene, the place of a gene, the position of a gene is what we call a locus. We're gonna bring these things in terminologies, and then please don't forget to give a correct answer. So here is a chromosome. A chromosome is here, but you have different parts, different locations of these chromosomes. So these parts is what you call the alleles. So these alleles may be for height, for size, for color, for blood group. They are supposed to be found on the same position of the chromosome. This side and this side is the same thing. So we call them alleles. So are alternative forms of gene. Then we talk about a dominant allele. So because you have two forms of alleles, one can be dominant, another can be recessive. And both of them can be dominant, or both of them can be recessive. So when we talk about dominant, it means something which suppresses another thing. Yes. So when we say that an allele that is expressed in the phenotype when found in the heterozygous state. When we talk about heterozygous, it means in different phases. One is like this, one is like this. So it means that this capital letter suppresses this. That's why I say that a dominant allele is that one which suppresses another allele. So this capital letter will suppress the small letter. Then what you call the recessive allele. This one can only express itself in absence of a dominant allele. So if this one is absent, then automatically this small t will also express itself. So we are saying that an allele that is masked in a phenotype when found in a heterozygous state. So it can only show itself in a homozygous. It is only expressed in a homozygous condition or homozygous state. Here is an example. You have homozygous dominant. It means that, you see, they are the same. Homo, I say that when you talk about homo, means the same. Zygous comes from what? Zygote. So it means that this zygote has the same kind of allele. Heterozygous, it means that this zygote has different kinds of alleles. So you have capital letter and small letter. And then homozygous, it's still the same type of alleles, but these alleles are recessive. So it means that it is the same kind. Homo, which means the same. So same, but they are supposed to be recessive. Then when you go to phenotype, when you talk about phenotype, it means the physical appearance of an organism. You are expressing an individual in terms of what you see. In most cases, when you bring a question concerning about a cross, they're going to ask you the phenotype. The first question, the first part, you are supposed to write the phenotype. We get these answers from the question. Is it clear? So we are saying that physical appearance of an organism is determined by the genotype. For example, tall or short. You can be tall, you can see that a person is tall. You can be short, you can see that a person is short. You can be light-skinned, a person is light-skinned. You can see them. And then what about genotype? Here you are describing an organism in terms of what? In terms of genes, genotype. The gene type. Which kind of gene type do you have? So here is genetic composition of an organism. So you know you are tall, but which kind of tallness? You might be tall, but homozygous tall or heterozygous tall. You have an allele for shortness. You can be short, but you have an allele for tallness. You see two people who are short, they produce a tall kid. Two people who are dark-skinned, they produce a light-skinned kid. Two people who are light-skinned, they produce a dark-skinned kid. And then a guy can say that, yeah, this is not my kid. It's whose kid? All right. So here is another example. We'll talk about this. Describing this in terms of the genes is what called the genotype. Then also this is the genotype, and also this is what? Genotype. So I will say that this is homozygous dominant, this is heterozygous, and then this is homozygous recessive. So I'm describing these flowers in terms of genetic composition. So then when we talk about homozygous, I've been talking about this. Homo, which means the same zygous from the zygote. Then it means that this zygote has the same kind of genes. Two identical alleles for a particular characteristic. Homo, same, can be homozygous dominant or homozygous recessive. When we talk about hetero, when you say hetero, it means different. So the zygote has different kinds of alleles. For example, capital letter B and then small letter b. So they are different letters or different alleles. If you look at this picture, you'll find out that homozygous, you have this. They are found on the same locus, same location. This one and then this one, and then this one and this, you see. While if you look at heterozygous, same location but different letters. Same location but different letters. So this is homozygous recessive, this is homozygous dominant, and then this is heterozygous. For the case of heterozygous, we don't have recessive and dominant, just say heterozygous. A monohybrid cross is a cross which involves one character. You are right, it involves one character. But now you said pink and white, it still is fine. The answer was correct. But where is the character there? The character is color. That's the character. But this character has two alternative forms. The blue you talked about and the white you talked about. It's either blue or it's white. So those are the what called the alleles. So the character is the color of that individual we talked about. And then alleles are the different types of colors you have talked about. You understand? Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel. You're saying is a cross which involves only one characteristic or trait. Trait is the same as the character, and it's being shown in the genetic cross. This guy Mendel put forward the law of segregation. He said segregate means that now individuals during meiosis, they are supposed to segregate, they are supposed to divide, so that the offspring must receive one from the parent male and one from the parent a female. So how do they form these gametes which are being formed, which are being given to the offspring? They're being formed during the process of segregation. So he says that diploid, it means that the organism has two sets of chromosome organisms. For example, how many chromosomes do you have? You have 46, but you have 23 pairs. So if there are 23 pairs, then we say that you are a diploid or diploid. Randomly, you don't have a choice which one is going, which one is not going. It's randomly to the offspring, such that the offspring receives one allele from each parent. The characteristics you have now, the parent gave you, each parent brought one allele. If you are tall, for example, it means that those genes you have, one parent gave you one allele for tallness, and another parent gave you one allele for tallness. So if both of them, they gave you a dominant allele, then it means that you are homozygous tall. If one gave you a dominant allele and another one gave you a recessive allele, then it means that you are heterozygous tall. So that is the meaning, that each parent is supposed to give you one character. Let's look at how do we mark. This is how we mark in exam. And if you miss something, then it means that you won't get a mark. Number one, whenever they give you a genetic cross, this is what you are supposed to write. P1, which means parent one, phenotype. Phenotype, physical expression of an organism. We get this from the question. A man who is dark-skinned marries a lady who is light-skinned. Dark-skinned man, light-skinned lady. That's it. That's the phenotype. So you go ready. Then genotype. Now, who is this dark-skinned, light-skinned? Is that person homozygous or is heterozygous? That is the genotype. So when you read the story they have given you, you'll find out that you'll be able to scoop out what is called the genotype. So we give you a tick here, we'll give you a tick here. From there, because you have now the genotypes, you have the genes for each parent, then meiosis must take place, segregation must take place. When it takes place, yes, you form what you call gametes. And then when these gametes, now you have, for example, the sperms this side, you have the ova this side, or the ovum this side, then fertilization must take place. Fertilization must take place so that offspring can be formed. Then you have F1, which means first filial generation. And then now you get the phenotype of the F1. I'm going to explain this. After that, you need to get what called the ratio, or the genotypic ratio, or the phenotypic ratio. Then you use the genotype to obtain the phenotype. Use the genotype, description of an organism in terms of genes, and then after that you'll be able to get what you call the phenotype. Writing the word meiosis and fertilization will also give you a tick. If you don't write it, you lose that tick. So now you have the gametes. These ones you bring them here, and then these ones you bring them here, or you can swap them. So that's how we do mark. How do we mark genetic cross? That's how I've been trying to explain. This is a practical example. So P1, F1, we give you a tick. Meiosis and paralyzation, we give you a tick. Writing the phenotype, we give you a tick. Writing the genotype, we give you a tick. Then crossing them, putting the gametes here, and then you cross them correctly, we give you a tick. Writing the phenotype, the genotype, we give you a tick, and then writing the phenotype, we give you a tick. P1 and F1 can change to P2 and F2 if it is the second generation. So now that's it. Another thing which we also do when we are marking you, yes, we make sure that there is a compulsory mark in the genetic cross. If they ask you a question, maybe P1 and F1, it should be the compulsory mark. And you have, this question is how many marks? For example, there are 6 marks. And then if you count here, 1, 2, 3, 4, 5, 6, 7, there are 7 marks. And then they say that P1, F1 is the compulsory mark. So you get the rest of these marks. If you count them, they are 6. We won't give you 6. We will give you 5. Why? Because you did not get this one correct. Maybe you only wrote P1 and you didn't write F1. So you didn't get this one correct. So when you are counting, we first count the compulsory mark and we count the rest. So make sure that when you are crossing it, at least cross it and maximize all the marks. Okay, let's look at dominance, which are the types of dominance. We can go through them very fast.
Welcome back to our class. Don't forget to subscribe if it is your first time to visit our channel.
We have 3 types of dominance: complete dominancy, codominancy, and incomplete dominancy. So let's look at them in detail. When we talk about complete dominancy, when you are dominant, it means that you have powers over another individual. So it's a genetic cross where dominant allele masks, blocks the expression of the recessive allele in a heterozygous condition. So in complete dominancy, you have heterozygous condition. So you have what you call the incomplete. When we talk about incomplete, it means that yes, the dominance is there, but it is incomplete. So it means that one brings 50 and then another one brings also 50. So you're saying that it's a genetic cross where none of the allele is dominant over the other. Neither of the allele is dominant over the other. It means that they have equal powers. The president of South Africa and the president of America, both of them, they have the same powers in their countries. So it means that neither of the president is dominant over the other. When it is the meeting, the president of South Africa will have the same contribution like the president of America. That's the meaning. Not that, no, because America is a superpower, now South Africa, no. Both of them, they are presidents. Neither of the two is, they are dominant, yes, they are dominant, but neither of them is completely dominant over the other, over each other. Although your country is big or small, still you have the same power as presidents. For example, you have white and you have white and red. When you cross them, they form what called the intermediate. Intermediate means that neither of the two is dominant over the other. When you cross them, they produce the intermediate. It's not white, it's not red, but it is in between there. It produces a new character. So the character which is being produced is new. That's the meaning. Yes. But here you have the one character from the parent. Black and white, you either produce white or you produce black. Here when you cross, you don't produce white, you don't produce black, but you can produce gray, which is in between the two. It's not white, it's not black, but is in between. That's the meaning, intermediate. And then you have the co-dominant. Co means together.